Hello friends, welcome back to our channel, and today we are discussing classification of bacteria. What is a bacteria and what is the characteristics of bacteria and what is the feature of bacteria we can discussing on this video. So let's start. Bacteria can be classified into various categories on the basis of features and characteristics. The classification of bacteria is mainly based on the following are shapes, composition of cell wall, mode of respiration, mode of nutrition on the basis of Fage law. Sixth one is on the basis of spore, on the basis of morphology, on the basis of salt concentration, on the basis of optimum pH for the growth, and last one is on the basis of optimum temperature requirement for the growth. This is the 10 classification of bacteria. Move to the first one is the classification of bacteria on the shapes. They are divided into bacteria shapes are three types, which is the first one is a coccus, which is a spear in shape. Second one is a bacillus, is a rod in shape. Third one is a spirillus, which is a spiral in shapes. Move to the types of cocci bacteria. First one is a monococcus. Second one is a dicoccus in shapes. Third one is a tetracoccus. Fourth one is a saccharina. Fifth one is a streptococcus, and sixth one is a staphylococcus. This is the types of a cocci bacteria. Move to the types of bacilli. First one is a single bacillus, which is a Bacillus cereus. Second one is the streptobacillus, which is a Streptobacillus moniferium. Third one is a Palisades. Fourth one is a Dibacillus. Move to the types of spiral bacteria are Vibrio. Second one is a Spirium. Third one is a Spirochest. Move to the classification of bacteria on the composition of cell wall are peptide gland cell walls, which is a gram-positive bacteria, and polysaccharide cell wall, which is a gram-negative bacteria. This is a difference between gram-positive bacteria and gram-negative bacteria, which is a polypeptide glycan is a in gram-negative bacteria is more than the gram-negative bacteria. Move to the classification of bacteria on the basis of mode of nutrition are autotrophs and heterotrophs. Move to the difference between autotrophs and heterotrophs are the organism that obtain carbon from the carbon dioxide and use to light energy in inorganic chemical compounds to produce complex organic compounds are known as the autotrophs. These are bacteria that can be synthesize their own food from the inorganic compounds. Examples are which is a photosynthesis, purple sulfur bacteria, and chemosynthesis with the nitrifying bacteria. Move to the autotrophs, which heterotrophs are the organism that can be make their own food but intends obtain nutrition from another organism source that may be live or dead. The example are the saprophytes, which is a bacteria of delay, and parasite, which is a streptococcus. Move to the classification of bacteria on the basis of optimum temperature of growth are they are divided into five categories. First one is the saprophytes, second one is the psychrotrophs, third one is the mesophiles, fourth one is the thermophiles, and fifth one is the hypothermophiles. Move to the difference between these five categories of are first one is the saprophytes, bacteria that can grow at 0 degrees Celsius or below, but optimum temperature of growth is 15 degrees Celsius for below and maximum temperature is 20 degrees Celsius are called sacrophiles. The examples are Vibrio, Psychrotheras, Vibrio marinus, Maphromonas and vacuolates and sacrophiles. Move to the cytotropes. Those bacteria that can grow even at 0 degrees Celsius but optimum temperature for growth is 20 degrees Celsius to 30 degrees Celsius. Move to the mesophiles. Those bacteria that can grow best between 25 to 40 degrees Celsius but optimum temperature for growth is 37 degrees Celsius. The examples are E. coli, Salmonella and Streptococcus. Move to the thermophiles. Those bacteria that can be best grow above 45 degrees Celsius. The examples are Streptococcus thermophiles, Bacillus and Streptococcus thermophile aquatics. Move to the hyperthermophiles. Those bacteria can have optimum temperature of growth above 80 degrees Celsius. The examples are Thermosulfobacterium and etc. Move to the classification of bacteria on the basis of optimum pH of growths. They are divided into three categories. First one is acidophiles, second one is alkalinophiles, and third one is neutrophiles. First one, acidophiles are those bacteria that grow best at acid pH. The cytoplasm of these bacteria are acid in nature, and some acidophiles are thermophile in nature. Such bacteria are called thermophiles. An example are Thiobacillus, Thiobacillus phaerodiones, and thermoplasm and sulfobacillus. Move to the alkalinophiles. Those bacteria grow best at alkaline pH. An example are Vibrio cupellina. An optimum pH of growth is 8.2. Move to the neutrophiles. Those bacteria grow best at neutral pH 6.5 to 7.5. Most of bacteria grow at neutral pH. An example are E. coli. Move to the classification of bacteria on the basis of gaseous requirements are aerobic and anaerobics. First one is aerobic, which is obligate aerobus. Those bacteria that can require oxygen and cannot grow in absence of oxygen. These bacteria carry out only oxidative types of metabolism. The example are Mycobacterium and Bacillus. Move to the aerobic. They are divided into four categories. First one is a facultative aerobus. Second one is a allotolerant aerobus. Third one is a microaerophiles. Fourth one is a obligate anaerobes. Move to the facultative anaerobes. Those bacteria that do not require oxygen but can use it if available. The example are Koki and Salmonella. Move to the aerotolerant aerobes. Those bacteria do not require oxygen but can tolerate of presence of O2. The example are Lactobacillus. Move to the microaerophiles, with those bacteria that can grow low concentration of oxygen but at atmospheric level of oxygen growth this bacteria is inhibited. The example are Campylobacter. Move to the obligate anaerobes. Those bacteria that can grow only in absence of oxygens are Streptococcus, and Methanococcus. Move to the classification of bacteria on the basis of salt concentrations are they are divided into two types, which is the halophiles and halotolerants. They are divided into two types, which is the halophiles. Those bacteria require a high concentration of HCl of growth, and cell membrane of halophile bacteria is made of glycoproteins with high concentration of negative charge. which is glutamic acid and aspartic acid. So higher concentration of En plus ions with concentration is required to shield the negative charge. The examples are Aerobacteria, Halobacterium, and Halococcus. Move to the halotolerance. Most of bacteria is not required HCl, but can be tolerant low concentration of HCl in growth media are called halotolerants. Move to the classification of bacteria on the basis of flagella are monotrichous bacteria. Second one is the locotrichous. Third one is the amphilotrichous bacteria. Fourth one is a peristicus bacteria, and fifth one is a asticus bacteria. First one is a monotrichous bacteria, the bacteria having single flagella in one end of cell. The example are Vibrio cholera and Pseudomonas aeruginosa. Move to the Lophotrichus bacteria, which is bacteria having bound of flagella in one end of cell with Pseudomonas fluorescens. Move to the Amphitrichus bacteria, which is bacteria having single cluster of cell of flagella but both end cell. This example are Aquaspirium. Move to the Peristicus bacteria, which is bacteria having flagella all over the cell surface, which examples are E. coli and Salmonella. Move to the Asticus bacteria, which is bacteria without flagella, which is a Shigella. Move to the classification of bacteria on the basis of spores, which is a spore-forming bacteria and non-spore-forming bacteria. The spore-forming bacteria, those bacteria that produce spore during unfavorable conditions, they are divided into two categories, which is endospore-forming bacteria and exospore-forming bacteria. Endospore-forming bacteria, which is examples are Bacillus, Clostridium, and Sporosaccharina, etc. And exospore-forming bacteria, which is the Methylocyanus. And non-spore-forming bacteria, which those bacteria which do not produce spore, which E. coli and Salmonella. So join our app, which is available on Play Store and which is linked in description. So subscribe our channel, share with your friends. Thank you.